Hello, my name is Ali Hamade, and in this presentation I will be explaining the mitochondrial machineries of the presequence pathway for protein import and assembly. My goal is to take this complex system and make it clear through visual representation. Unless otherwise noted, all of the animations in this presentation were made using Chimera, a 3D molecular modeling software. Disclaimer, some of the systems discussed here are still not fully solved, and so some models shown are incomplete and are for visual purposes. Mitochondria are most commonly known for their role in ATP energy production. However, mitochondrial proteins also play a crucial role in metabolic and regulatory functioning in eukaryotic cells. More than 98% of mitochondrial proteins are synthesized as precursor proteins in the cytosol, the intracellular fluid inside the cell, and then transferred to the mitochondria via specified pathways. Scientists used to believe that there was only one main pathway for all precursors. However, now we have discovered there to actually be 5. Not only are there 5 pathways, but they are also interconnected and share systems of transport. It is important to understand the different pieces of each pathway in order to prevent illness associated with their defects, such as diseases of the nervous system, heart, and muscles. I will be going through one of these systems, the first and most used pathway, the presequence pathway. Beginning in the cytosol, about 2/3 of the mitochondrial proteins are synthesized with a cleavable N-terminal presequence that is used as a targeting signal tag by certain receptors and translocases, namely the TOM and TIM23 complexes. This presequence is then cut off of the precursor after processing by a peptidase. Presequence precursors can range anywhere from less than 10 to 100 amino acid residues. They have a tendency to form amphipathic alpha helical structures. This results from their hydrophobic face and a positively charged residue face interacting with the receiving TOM complex. As shown here in this presequence peptide, one side is rich in hydrophobic residues such as leucine and alanine, depicted in red, and the opposite side consists of positively charged residues such as arginine, depicted in blue. The first complex that the presequence precursor enters is the translocase of the outer mitochondrial membrane, or the TOM complex. This 148 kilodalton symmetrical dimer complex consists of the core beta-barrel protein TOM40 surrounded by alpha helical subunits TOM5, TOM6, TOM7, and TOM22, and is interconnected with proteins TOM20 and the slightly distant TOM70, which are responsible for stability and for being receptors of incoming precursors. The image shown here is a combination of 3 solved structures to form a 3-dimensional visualization of the whole TOM complex. TOM20 is the first receptor the precursor interacts with and is responsible for the recognition of the hydrophobic surface of the presequence. It consists of 5 alpha helices and a tetracopeptide repeat structural motif, or a TRP, that preferentially folds to produce a domain with an internal hydrophobic core. This core forms a central groove that consists of side chains that are shown here in green. The hydrophobic presequence side, in this case consisting of leucine 4, 7, and 8, shown in red, is bound to the groove to initiate import of the precursor. The TOM complex has a main protein called TOM40. This is a beta-barrel protein consisting of 19 transmembrane beta strands. It is responsible for the translocation of preproteins through the outer membrane, as well as sorting the different precursors upon entry. TOM40 is presumed to be a voltage-gated ion channel, which forms the central pore of the protein import machinery. This is involved in more than one import pathway, as the center consists of both hydrophilic and hydrophobic regions that interact with a variety of preproteins. TOM40 is assembled and stabilized by the small subunits TOM5, TOM6, and TOM7, as color-coded here, that interact with TOM40 beta strands 10 through 11, 13 through 15, and 3 through 6. TOM22 protein is a transmembrane helix centrally located between 2 TOM40 promoters that recognizes the positively charged backside residues of precursors and binds them to the complex. It does this via its negatively charged N-terminal. It is also the key stabilizing factor in the linkage between the two TOM40 barrels through its interaction with strands 1 through 3 of one promoter and with strands 16 through 19 of the other. This stability protein is so important that the deletion of TOM20 receptor has been shown to be lethal. In the animation, van der Waals interactions between TOM22 helices and the respective strands mentioned can be seen through visualization of just a few of the short distances between the residues. These interactions can also be seen between the two protein surfaces. Lastly in the TOM family is the TOM70 protein. It is responsible for recognizing non-cleavable hydrophobic precursors, such as ones found in the carrier pathway. It consists of multiple tetracopeptide repeat motifs, which are organized into right-handed helices. Just like TOM20, TOM70 forms a peptide binding groove. Although this protein plays a larger role in non-cleavable proteins, it can substitute as a less efficient alternative for one of the other TOM receptors if they become defective. After the preprotein is translocated through the TOM complex, it goes to the translocase of the inner membrane, the TIM23 complex, which consists of a multi-subunit system containing proteins TIM50, TIM23, and TIM21. This translocation happens in the intermembrane space, where the TOM complex and the TIM23 complex are connected through the TIM21 protein. Among a few others, there is also a less understood TIM17 protein that is strongly linked to the regulation of TIM23 and to preprotein sorting in the inner membrane. This complex translocates presequence preproteins through lateral sorting into the inner membrane via a membrane potential or directly to the matrix via ATP import motor PAM. Here, I have provided a simplified image of the primary structure of the central TIM23 protein to show that it is involved both intra- and extracellularly to the mitochondria. As expected, it has a large cluster of its structure in the inner membrane, where its main function resides. The green residues indicate N-glycosylation that helps localize the protein to the mitochondrial surface. TIM23 protein is the central component of the TIM23 complex. Its structure forms a voltage-gated ion channel through which precursor transport is driven. TIM17 is a protein that is tightly connected to the TIM23 protein. TIM17's function is still being studied, however, it seems to be involved in the regulation of the TIM23 channel. It is directly connected to TIM23 and is identical in structure. TIM23 protein consists of 4 alpha helical transmembrane segments that are embedded in the inner membrane and a hydrophilic domain facing the intermembrane space. An exact TIM23 PDB crystal model has not been developed. For the purposes of this presentation, the images provided show a TIM23-like structured protein based on the ADP-ATP carrier AAC2P. Its GXXXG structural motif influences hydrophobic interactions in the inner membrane space. This is presented in the animation through some of the hydrophobic residues shown, as well as the top-down view of the 2 helix strands from the animation as helical wheels. This arrangement is crucial for the structural integrity of the protein. In the hydrophobicity surface image of the protein on the top right, you can see the hydrophilic region that interacts with the intermembrane space. The mitochondrial membrane potential plays some key roles in preprotein processing. It acts on the positively charged presequences to help transport them through the inner membrane, allowing for less energy usage. It has also been shown through research done on membrane potential alteration that structural proximity of the proteins within the TIM23 complex is dependent on the membrane potential. This proximity determines the functionality of the proteins. Therefore, the condition of the membrane potential affects the structure and function of the TIM23 complex in the inner membrane space. The structural effect is shown in the regulation of the TIM23 membrane channel by TIM50. Normally, the TIM23 channel is open, but the attachment of TIM50 regulates it so that it is only open when a presequence is bound. TIM50 is the presequence receptor that binds incoming preproteins in the intermembrane space. It is largely in control of maintaining the permeability barrier of the mitochondrial inner membrane. Furthermore, a membrane protein called MGR2 binds to the hydrophobic sorting signal of the entering presequence at the TIM50 and controls the preprotein release into the inner membrane. MGR2 is also known as the lateral sorting gatekeeper and functions as a quality control of entering precursors. In the animation, a positively charged presequence peptide forms an ionic bond with TIM50 at the binding site, formed by the negatively charged residues. TIM50 then triggers TIM23 to open and become active through its linkage at the protruding beta hairpin arm. MGR2 has been omitted from the animation for the sake of simplicity. The close proximity between the binding site of the presequence and the binding site of the TIM23 is likely related to the role the presequence has on the channel activation. TIM21 is a protein subunit that connects the TOM complex and the TIM23 complex in the intermembrane space through its binding with the TIM22 complex. The TIM22 complex is involved with the carrier preprotein translocation pathway, however, is interconnected with the presequence pathway in this regard. TIM21 regulates how many preproteins interact with TIM50 to prevent accumulation. It also plays a role in the physical linkage of the TIM23 complex to the respiratory chain 3 and 4 supercomplexes. This linkage allows for a more efficient interconnected system as the respiratory chains act as a platform for PAM subunit assembly. What is interesting about the TIM21 structure is that it has a unique alpha-beta mixed protein fold. The three-dimensional structure of TIM21 is composed of 2 alpha helices that are surrounded by an 8-stranded beta sheet, all connected by loops. It is also strongly hydrogen bound to the water of the intermembrane space where it is mostly located. After intermembrane translocation, import into the matrix is promoted by ATP energy that is provided by the presequence translocase associated motor, PAM. The PAM complex is located on the matrix side of the mitochondrial inner membrane. The core of the motor is an ATP-driven molecular chaperone that is a heat shock protein called MTHSP70. MTHSP70 is not only the main part of the ATP-produced import of preproteins, but it is also involved in many import bodily processes, such as mitochondrial homeostasis, protein folding, and even in cell death. This importance has caused the MTHSP70 to develop another name, mortallin. The mortallin structure is split into 4 subdomains. Its substrate binding site consists of a beta sandwich domain specific for basic hydrophobic sequences and an alpha helical top domain. The binding pocket is occupied by water molecules and a sodium ion that are responsible for maintaining closed conformation for crystal packaging through a salt bridge. This chaperone protein is recruited by a peripheral membrane protein called TIM44 that couples it to the TIM23 channel. There are many more proteins that support this system. To name a few, PAM17 is also involved in the linkage of TIM23 to mortallin. The ATP hydrolyzing activity of MTHSP70 is regulated by stimulatory and regulatory proteins PAM16 and PAM18. And there are many other chaperones that help prevent misfolding and aggregation of proteins in this PAM complex. Among these is a very important one called ZIG17. HSP70 or mortallin likes to self-aggregate, causing biogenesis failure, and so it has a co-chaperone, ZIG17, that specifically prevents this. ZIG17 is crucial for keeping the mortallin functional. ZIG17 forms an L shape and has 2 zinc finger motifs consistent of 4 cysteine residues localized around the zinc molecule at the end of the L. The zinc is surrounded by 2 two-stranded anti-parallel beta sheets, shown in green, and shown in red is the top of the L that has 2 alpha helices intertwined with a single beta strand. The concaved region around where aspartagine 111 would be forms the binding site on TIM17 for mortallin. The precursors cannot become mature proteins until the presequence tag is removed. This is the job of the mitochondrial processing peptidase. The MPP is a heterodimeric peptidase located in the mitochondrial matrix that degrades presequences. This degradation can take place during or after the transport through the TIM23 complex. Some intermembrane preproteins need to be cleaved twice, so after the MPP, the intermembrane peptidase cleaves the sequence again. MPP is made up of two heterodimeric subunits that each contain two sets of 3 alpha helices packed against 6 stranded beta sheets with 5 other alpha helices at one end of the sheet. The domains are connected by a 23 residue linker. This peptidase also has a zinc metal ion that is localized in the cavity of the active site. It is coordinated by beta-histidine 70, beta-histidine 74, and beta-glutamic acid 150, which are involved in both the zinc binding and catalysis. As we've discussed, presequence preproteins are synthesized in the cytosol, translocated through the outer membrane by the TOM complex, through the inner membrane by the TIM23 complex, and are then cleaved by the MPP. Finally, the mature protein is then ready to be sorted and exported into the mitochondrial inner membrane. This is either done laterally by TIM23, as mentioned earlier, or by the oxidase assembly translocase. In the provided protein string model, we can see in a web-like system some of the different protein complexes and their connections with each other to make the whole presequence pathway. Here are my references, and thank you for watching.